And continue on with bone structure and function. The two overriding functions served by bone are as a structural support for the skeleton and as a reservoir for calcium and phosphorus. Recognizing that the organism depends on calcium as a second messenger and on phosphorus as a carrier of energy, ATP and GTP, and as a component of nucleic acids, it is a fortunate result of evolution that both the mineral bank function of bone and the support function were met by the development of the calcium phosphate-based skeleton. If bone is to serve as a reservoir for calcium and phosphorus, there must be a mechanism for removal and replacement of bone tissue, which is formed in the extracellular matrix. Bone is a specialized connective tissue in which the extracellular matrix is composed of crystallized mineral salts, 50%, collagen and glycoproteins, 25%, and water, 25%. Four types of cells are found in bone tissue: osteogenic cells, osteoblasts, osteocytes, and osteoclasts. Osteogenic cells are stem cells derived from the mesenchyme. When osteogenic cells divide, they form osteoblasts. Osteoblasts are bone-forming cells that synthesize and secrete collagen and other organic components into the extracellular space, and which initiate calcification. Osteoblasts derive from a pool of mesenchymal precursor cells in the bone marrow. During differentiation, osteoblasts express parathyroid hormone and vitamin D receptors, and express the enzyme alkaline phosphatase. Osteocytes are derived from osteoblasts that have become entombed in matrix secretions and crystalline matter. Osteocytes carry out the routine metabolic functions of bone, such as exchange of nutrients and waste with the blood. It is also proposed that osteocytes sense mechanical loading and secrete signals that initiate bone remodeling. Osteoclasts are responsible for resorption of bone and long-term maintenance of normocalcemia. They are very large cells that derive from multiple monocytes. Thus, osteoclasts derive from a hematopoietic precursor. On the side of the osteoclast that abuts the bone surface, the plasma membrane is folded into a ruffled border. It is at this site that the cell releases lysosomal enzymes, cathepsin K, and acid that dismantle the protein and mineral components of the adherent bone. As noted earlier, what makes bone different from other types of connective tissue is that the secreted extracellular matrix contains abundant inorganic mineral salts, primarily hydroxyapatite, which is formed from simpler precursor calcium-phosphorus compounds. These minerals are deposited in intimate contact with the collagen fibers, which are embedded in the extracellular matrix. There are minerals crystallizing, the minerals crystallize, causing the extracellular matrix to calcify. The interplay between the hardness of the hydroxyapatite and the tensile strength of the collagen fibers accounts for the remarkable strength of the bone while minimizing the weight of such a robust structure.